Today I want to talk to you about photorespiration. Photorespiration is something that happens because Rubisco evolved way back before there was oxygen in the atmosphere. So Rubisco is this enzyme that does the Calvin cycle. And so Rubisco takes inorganic carbon, right, carbon dioxide, and it adds it to an organic molecule to make another organic molecule. And so Rubisco evolved way back before photosynthesis was really a thing, right? It was really in the beginning of the evolution of photosynthesis. So when photosynthesis started, the planet had very little oxygen in the atmosphere. We needed a lot of cyanobacteria to be doing photosynthesis in order to get oxygen in the atmosphere. So Rubisco takes this RUBP and carbon dioxide, and it makes organic molecules. But Rubisco, sadly, can also use oxygen as a substrate instead. So Rubisco is the carbon fixation enzyme in the Calvin cycle. That's what it's supposed to do. It normally binds to the carbon dioxide. And so carbon dioxide is the optimal substrate along with RUBP. And it builds sugars, right? That's photosynthesis. That's what it's supposed to be doing. But unfortunately, Rubisco evolved when there was very little oxygen in the atmosphere. So if Rubisco, this enzyme that works with RUBP and carbon dioxide, if instead there's a lot of oxygen around, Rubisco can bind to oxygen instead of to carbon dioxide. And that breaks down glucose, that breaks down sugars, which is called photorespiration instead of photosynthesis. So here's RUBP, and here's oxygen, right? It's not supposed to be here, but it is because we have a lot of oxygen around now. So Rubisco, instead of using carbon dioxide, if it uses oxygen instead, if it uses oxygen as a substrate, that's actually going to break down this 5-carbon sugar and release carbon dioxide. So it's releasing carbon dioxide, not making any ATP, and not making any sugar. So remember, photosynthesis is supposed to make sugar by using carbon dioxide. Now instead it's breaking down sugar. So it's doing the exact opposite of what it's supposed to be doing. That's called photorespiration instead of photosynthesis. So the impact of photorespiration. Plants can lose 50% of their carbons that were already fixed by the Calvin cycle. So here's Rubisco. Rubisco is supposed to take carbon dioxide and produce sugar, right? That's what it's supposed to be doing, making sugar. But instead, if Rubisco uses oxygen as a substrate instead, it's going to break down that RUBP, releasing carbon dioxide, doing the exact opposite. So photorespiration makes no ATP, and it makes no glucose. Instead, it breaks down glucose. So that's bad stuff. So we don't know why, except that maybe Rubisco just evolved when there was no oxygen around, right? There's a little bit of evidence that maybe Rubisco, using oxygen, kind of protects the plants. Maybe photorespiration has a little bit of a protective adaptation, possibly, so that when plants get sort of like making too much ATP, maybe it sort of cools things down, but we're not really sure about that. It looks like it just might be a product of the evolutionary history of Rubisco, that it evolved when there was no oxygen. So evolution just tinkers with what's already available. So even if it isn't super efficient, that might actually be the reason that Rubisco does photorespiration. So for many plants, photorespiration is a problem. So if a plant evolves in a place where, or if a plant lives in a place where it's really hot and dry a lot, photorespiration is a big problem, because what happens is those stomata will close up because the plant is losing too much water. And when the stomata close up, right, here's the stomata. They're supposed to be open. When the stomata close up, now at least the plant's not losing water, right? Because in the stomata, if they're open, the water will be lost. But the problem when the stomata are closed is that carbon dioxide can't enter either, right? So carbon dioxide is supposed to enter, oxygen is supposed to leave. If the stomata are all closed up, water's not leaving, but oxygen is also not leaving, right? So if oxygen's not leaving and no carbon dioxide is coming in, then Rubisco is going to have more oxygen to use and less carbon dioxide to use. So the plant's going to be doing a lot of photorespiration, kind of a bummer. So if that happens a lot, there's a strong selective pressure to evolve something different. Anything that a plant can come up with that's different is going to be selected for. So there are three types of plants that I want to talk about. The C3 plants we've already talked about. C3 plants are the normal ones that just take this carbon dioxide. Rubisco will take that carbon dioxide with the RUBP and make these 6 carbon molecules that break down right away and make this 3 carbon molecule. We're going to call those C3 plants. Those are kind of the normal ones. So to reduce photorespiration, there are some plants, like certain grasses and cacti, that have adaptations to sort of make photorespiration less, to decrease the amount of photorespiration. So there are C4 plants, and then there are CAM plants. So I'm going to talk about each of these. These would be CAM plants right here. C4 plants are grasses. C4 plants will physically separate out Rubisco from the light reactions, and CAM plants will separate them by time of day. So that won't really make sense till I show them to you. So let's look at those. So here are C4 plants. So they'll physically separate out the light reactions from the Calvin cycle. So let's say the light reactions are happening over here in this cell and lots of oxygen is being produced. So if you have lots of oxygen, you remember Rubisco is here doing the Calvin cycle. If you have lots of oxygen, then Rubisco will use the oxygen instead of the carbon dioxide and break down sugar. Not a good thing. So these are physically separated out. The bundle sheath cells are where Rubisco will happen, where the Calvin cycle will happen. And then in these mesophyll cells, the light reactions will happen, which will produce lots of oxygen. And there's another enzyme, PEP carboxylase, which will actually fix carbon dioxide instead. It'll make this molecule called oxaloacetate. I'm not even sure that I'm pronouncing that the right way, which is converted to malate. Malate physically will be what's brought into these bundle sheath cells. The malate will get broken down to make carbon dioxide. And then Rubisco here has lots of carbon dioxide, not a lot of oxygen, and that will refix the carbon dioxide for a second time. So that just physically separates out the light reactions with lots of oxygens from Rubisco. So you have this other form of fixing carbon. So you might say, why? Well, a couple questions here. Why not just let PEP carboxylase do its thing? Why bother with Rubisco at all? And all I can say with that is that evolution works based on what's available. And the glucose that the plants are going to use to store their carbon is just made here with the whole Calvin cycle. And this other molecule, this other enzyme here, doesn't end up making that. And so this is just the best thing that plants have come up with so far to deal with Rubisco's problem, right? With Rubisco's problem with oxygen. So it's not crazy efficient, but it's what plants do. The other question you might ask is why all plants don't do this. How come it's just the grasses like corn and stuff that do that? Why not every plant, why not every plant out there? How come this hasn't taken over? And the reason for that is it actually takes a little bit of ATP to shuttle these molecules back and forth. And so there's a cost to this. It's worth it if you live where it's really hot and dry, but not worth it in every environment. So this is a selective advantage in really hot environments, but in environments that aren't so hot, where plants have plenty of water, this would be a selective disadvantage, right? Because it requires some ATP to do this. So C4 plants, you have this different enzyme, PEP carboxylase, has a better affinity. Affinity means how closely the enzyme attaches to its substrate. So it has a better affinity to carbon dioxide than it does to oxygen. It sticks to carbon dioxide better. So it's a nice adaptation for hot, dry climates. This would be selected for if it's really hot and dry. They have to close their stomata a lot if it's hot and dry. When plants close their stomata, they're not losing so much water. Yay! But they're not getting carbon dioxide, so the carbon dioxide is going to go down, and they're not releasing their oxygen. I know these things can go through lipid bilayers, but they don't go through the waxy layer here on the plant's leaves. So if Rubisco is exposed to lots of oxygen with less carbon dioxide, it's just going to do a lot of photorespiration. So this is an adaptation to that. So this is in sugarcane. You don't have to memorize that, certain grasses. It takes energy to ferry the malate back and forth, so only about 3% of plants on the planet are these C4 plants. There's another adaptation called CAM plants, crassulacean acid plants. Don't memorize that for sure, or even canned plants, but just that there's this other adaptation. So this is true for succulents and cacti and stuff. Okay, so what happens is that during the day, the light reactions happen, and PEP carboxylase, again, same enzyme, right? So there must be a common ancestor between the CAM plants and the C4 plants. Anyway, they'll take the carbon dioxide, use PEP carboxylase, and make that oxaloacetate and malate. Again, I'm not even sure if I'm pronouncing that one right, but they'll sort of do the same thing, and they'll do this stuff during the day, and then they'll just store the malate all day long. And then at night, so during the day, the stomata are closed, and Rubisco's not really doing much, and you just store this malate around. And that keeps a high concentration of carbon dioxide around. And so then at nighttime, do I have that? I don't know if I wrote that right. And then at nighttime, the stomata can close. So during the day, stomata close and release carbon dioxide from C4 plants to keep the Calvin cycle. Yeah. So at nighttime, the stomata are open, and the plants can then release the carbon dioxide, and Rubisco can do its thing. So at night, the open stomata will fix the carbon dioxide from this malate. So the malate will release the carbon dioxide, and then the Calvin cycle can work. So this is what happens at night. Okay, so the CAM pathway also requires ATP, and so not every plant does this. Only plants that have to evolve in places that are really, or that only in plants that have evolved in places that are really hot and dry, like deserts. So it's an adaptation that's really helpful in some places, but not other places, because it does take some ATP to do that. So the big picture here is really that there's some diversity in plants, and some adaptations are selected for in some environments, but not in other environments. So around here, there's not so many of these plants because it takes some energy to do this step. So C3 plants really have more of an advantage in places, where we live. My students in Massachusetts. All right, that's my story.